So I'm Lauren Zazadel. I work in the Amman lab at the Koch Institute at MIT. So I'm looking for a way for the immune system to detect aneuploid cells and clear them from the body before they do anything like turn into cancer. Aneuploid cells are cells that contain an abnormal chromosome content.
So I'm studying aneuploidy using mouse models that have mutations in them that cause persistent chromosome desegregation. So there was a surprising discovery just before I joined the lab that one of these aneuploid mouse models that we work with actually has tissues that are very euploid, and so they have the correct number of chromosomes. So one hypothesis is that the immune system could be clearing these cells. So in trying to solve the mystery of why we don't have aneuploid cells in this mouse, I'm taking a sort of whole body approach where I'm looking through all of the tissues and look for systemic changes that could be clues to what's going on and what the altered state is in this mouse that is causing the elimination or selection against aneuploid cells. Well, this image actually has 363 different images of mouse tissues, and this covers nearly all the tissues in a mouse actually. Each brick is a different view. The blue is structures that tend to be more acidic, such as the nuclei in cells, and then structures that are pink tend to be more basic, such as the muscle and red blood cells. These images were all compiled while I was looking through the whole body of this mouse, looking for changes in the presence of aneuploidy and clues as to the elimination of aneuploid cells. The tissues were chosen partly on visual appeal as well as ones that showed the phenotypes in my mouse. There's a lot of variety in these tissues, but one of the things that I have to do is find some observations that are relevant to my project and then narrow down my path. I have found one that I want to follow up on. So I'm looking for things like sign of inflammation, differences in tissue structure, any different disease state, or particularly the signs of inflammation would show me that there's increased immune activity in those tissues. Aneuploidy is very commonly found in cancer, and so if I find a mechanism that the immune system can use to eliminate these aneuploid cells before they turn into cancer, then this would also let us know what the aneuploid cells have to overcome in order to persist in the tumors. Part of the challenge of working with all of these tissues is excluding the ones that are not going to be relevant to the question I'm asking right now. I like the variability in day-to-day bench work in doing science, and the fact that the question and what you're doing is sort of constantly changing and adapting based on what you find, so it never gets boring. This took about 2 weekends, very intense weekends.